I remembered. Woo. There'll be an updated syllabus on Canvas. I'm going to try and post that after class. Just, excuse me, for a variety of reasons, we're, just slightly off on our current schedule. So we'll probably be shifting things a little bit, but still we'll finish in a good spot. We're going to be shifting to microbial pathogenesis pretty soon. So, I mean, is there a cooler topic? I don't know. I genuinely don't know. There's board games, but then there's microbial pathogenesis. So those are the top. They're my top probably three, and I can only teach one of those in this class. So pretty good. Pretty good. Also look out for an optional check-in survey. So this is just one more way that I'm going to be supporting the class and you individually. It will just give you some additional space to tell me how the exam went. Tell me if there are other things that I can do to help us finish strong, right? Like, we're still going to go through content and we're still going to learn things, and there's still one more exam, but that doesn't mean it has to be difficult and painful, right? I really want us to finish strong, and this is a way for you to share those thoughts. And then there are no student success hours. I moved those earlier in the week, as you probably all know. But you can always email me if you want to try and meet one-on-one tomorrow or next week. I think I still have 1 or 2 slots available, and then of course next week. So email, always a great opportunity. And then thank you if you have emailed me. I received quite a few, just generally, just sort of giving me some feedback or some positive comments. And sorry I haven't responded, but I really do appreciate that. I love, empowering students, and I'm glad that you've found that these efforts are connecting with you. Here are some great photos that I didn't get to squeeze in last week, but yeah, lots of diversity out there. Stationary, great lunch, and then I think the dog was like not happy with the owner or something. I forget, but anyway, yeah, lots of great photos. Sorry I can't include them all, but thank you for sharing a little bit more about your lives and your experiences. All right, so to the content. Are we ready, class? Are we ready? Or did we all fall asleep from mindfulness? I can't. Have I told you all that I love pirates, like like Pirates of the Caribbean and stuff? Like, so the IA captain thing, hilarious. Anyway, you forgot one very important thing, mate. I'm Captain Jack Sparrow. Oh my gosh, what an amazing line. Okay, anyway, trust me, I did not make too many SpongeBob references. All right, so I know last week we were lives under a pineapple under the sea. Okay, that's like pretty much all I know. Anyway, so last week we were talking about molecular clocks, and I felt like, for whatever reason, like we were not like on the same wavelength there. And so I really want to make sure we at least revisit this concept, because it is important as a framework to help us understand evolutionary forces that drive evolution, right? That drive evolutionary forces that drive how organisms will adapt to environments, thrive, survive, die off, right? This molecular clock is a way to sort of interrogate these relationships. The reason I laughed is because AZ did a Pinocchio face, and you know more. But I actually, yeah, there might be one other like random fact I know about like SpongeBob SquarePants, but that's about it. And so last time we spent a fair amount of energy sort of dissecting this sequence thing, right? And some of you were like, Dr. Adams, like, you can't just use the same slide to explain a concept that was confusing. And to you, I say, correct, correct. So I made a bunch of more new slides, and we're going to explore them together. And I'm taking an entirely different approach to hopefully connect this concept with the ideas of evolution and mutations. Okay, so forget molecular clocks for a second. We're just going to talk about some basic concepts in biology. All right, so we have this first idea, all right, that DNA randomly acquires mutations each time a cell divides due to errors in the replication process. Okay, so hopefully that's not like a news flash to anyone, right? It's like, whoa, whoa. No, like, I think we all are aware of this, so that's good. All right, so every time a cell divides, there are some random mutations that are acquired. And this DNA, right, is ultimately encoding proteins, right? DNA to RNA, RNA to protein. And so DNA is randomly accumulating these these mutations. At some point, that's going to affect things at the protein level. Okay, so three basic points here. DNA is randomly acquiring mutations. The DNA ultimately encodes proteins, and thus, if DNA is getting mutations, at some point, the proteins are going to get mutations. Okay, so now I'm breaking this down in this little model here. Okay, what we have in this model are a couple of things. This is me sort of explaining it in a visual sense. So for those of you who are like, oh, I love the visuals, great. This is for you. So we're just going to walk through this one step at a time. And some of you are saying, Dr. Adams, that's impossible. You drew too much. Like, this is not one step, this is many steps. And to you, I say, oh yeah, well, we don't want to do red. We're going to do white. All right, so we're just going to look at this first step, okay? You have a bacterial cell, the first bacterium, all right? And to be clear, this is a very simplified example, okay? This is very, very simple, but the concepts here are consistent with the main point, all right? So you have this first bacterium. It has 0 mutations, okay? It's the first one. It's just it just is, all right? So you have the first bacterium. That's great. And then you go to the next step, okay? And sorry, I know this is a little messy, but then you go to the next step, and the bacteria divides into 2, okay? Shoop, shoop. You see that? And it also makes that sound effect when it does it. It goes, whoop, whoop. All right, that's exactly what happens. And so during that division, a mutation was generated, okay? DNA polymerase, it's not perfect. Every once in a while, it's going to slip up, all right? I'm not perfect. Every once in a while, I'm going to slip up, all right? So the bacterial cell divided into 2. It's the oldest magic trick in the game, right? It's like 1, 2, right? You do that, your friends do it. It's like, hey, you want to see a magic trick, right? That's what bacteria is doing. Except it acquired a random mutation because it's not perfect. And this one also required a random mutation, but they're in different spots, all right? So one's over here, one's over there. Great. Why does this matter? Well, because it's going to keep replicating, right? It's going to keep dividing. It's going to keep accumulating these mutations. So you have these mutations here, you have these mutations there. Likewise, for this cell down here, you get another mutation up here and then another mutation down here, all right? But both of these two cells, okay, both of these two cells have the same mutation they inherited from the mother cell, okay? So you have the mother cell mutation right there, but then they both got new random mutations in different spots. And the same thing happened up here. You have this mother cell divides, and then this mutation, exact same, but this mutation is different, right? And then importantly, if you compare this cell to this cell, those are really far apart, right? This one has two mutations over here. This one has a mutation here and a mutation there. None of them are the same. And so what this means is as you keep dividing, as you keep replicating, you acquire more and more random mutations just by virtue of there being a slight error in your replication machinery, okay? So these mutations will just accumulate and accumulate generation after generation after generation. All right, does this make sense, everyone? I'm not moving forward till I get a yes, so. All right, crushing it. Okay. That's what I should do. It's like, if I really want participation, like I'm not moving forward until I hear a yes from someone. Okay, I think we're in a good place. So we're gonna move forward. Thank you. All right, so now we're gonna do that same concept, all right, except instead of just looking at this at the cellular level, we're looking at this at the protein level, okay? So instead of looking at that first bacterium, we're going to zoom in, all right? Oh, let's zoom. All right. We zoom in and then we see these random proteins, okay? So we see these random proteins. That's great, okay? And then we're going to zoom in even further. Look, we have this random ribosomal protein, okay? So that's why microscopes are cool, right? We can zoom in on things. We can scope out the situation, for example. And good one, Dr. Adams. Thank you very much. All right. So what scientists do is they look at DNA sequences that have to see how many mutations are present for individual proteins. So they're not looking at the entire DNA sequence for the whole organism. We're like just focusing on individual proteins. And so then they basically want to look at specific proteins that are conserved across all cells. All cells have these proteins, and they want to see how similar or different they are. All right. So that often means they're looking at a ribosomal protein, for example. All right. So we have a ribosomal protein. Great. Awesome. And so we have this ribosomal protein. And now we're going to look at the DNA sequence of the ribosomal protein. Okay. So it's the same process. All right. We have the first ribosomal protein from that first cell. It doesn't have any mutations because it's the first cell. It's the first ribosomal protein. It's fine. It's fine. It's perfect. Okay. This is the one perfect protein. Then there's replication, right? So the mother cell replicates, everything replicates. And now this protein, look, it's not perfect. All right. That is the protein. This is the protein version of Dr. Adams forgetting to press record. Okay. It's like, oh, made a mistake. There you go. It happened. Right. Or maybe it's this version. Dr. Adams forgetting to post the slides right after class. That's that version, right? Not perfect. And maybe I shouldn't point out all of my flaws, but I'm being real. Okay. So acquire these mutations, not the end of the world, but Now it's different. I'm different. I'm different. Okay, so this ribosomal protein is different, and it's also different from this other ribosomal protein because the mutations are different. Same process continues. Now you've acquired some more mutations, right? But let's see, this mutation, that mutation right there is this mutation. Oh, can't use the white circle. This mutation, all right. Boom. Boom. It's the exact same mutation. What's different are these mutations over here. This mutation is way over here. I don't see any mutation over here for this one, right? So instead, there's this mutation here, which should be there, okay? So now these proteins are diverging in their sequence, okay? The mutations are in different spots. The same thing is happening after one round of replication here, all right? So you have a different mutation, and now boom, look, it still has that one it inherited from the mother cell, right? But now it has a different one here, it has a different one here. And then you can do the same thing. You can start comparing these, right? These are somewhat similar because they both had the same mother sequence. And then these are somewhat similar because they had the same mother sequence. But these are super different, right? This is this is different than this, right? Because none of these mutations are the same. And so this is why it's so important to think about mutation rates during replication, because we can use this information as a tool to figure out how related everything is to each other, okay? So that is why we study these sequences and try to assess mutation rates, because using this information, we can then use what we call the molecular clock. The molecular clock is basically looking at these mutations in certain proteins that are shared across all different cells. And by looking at those sequences and how related they are to one another, we can understand which came first, how old something is relative to another one, and how genetically related those organisms are. Okay, so genes that show the most consistent measures of evolutionary time encode components of the transcription and translation apparatus. And the most widely used molecular clock is the gene that encodes the small subunit rRNA, or SSSU rRNA. That's the 16S in rRNA in bacteria, or the 18S RNA in eukaryotes. And the reason this is the most widely used one is because this is highly conserved, and it's found in all these different domains, and you're able to have consistency when trying to assess, hey, how conserved is this sequence relative to another species? And just by looking at mutation rate, you can start to understand these evolutionary relationships. And I know some of you are saying like, oh my gosh, like you're just making like random pictures. Where are the sequences? All right. All right. You want to see some sequences? I'll show you some sequences. Boom. Sequences. All right. So if we look at these sequences, I just added some sequences that are similar to the ones found in your book from that original schematic that we looked at last week. And we have this root sequence, okay? The root sequence is basically the original sequence, the one that you're saying was first. And you choose the root sequence. That is basically a way of trying to establish that original defining sequence that everything else is connected to, okay? So now this is the picture version. This is the, the sequence version of what's going on. So the original sequence, all right, is ATG, TTC, TT, okay? It could be anything, it doesn't really matter, but for the purposes of the simple example, that's the sequence. Then after one round of replication, this A was changed to a C. That corresponds to this mutation here. Meanwhile, the other cell that divided changed the T to a G, okay? So sequence-wise, there has been this change. Sequence-wise, there's been this change. And all the logic that we just discussed still applies. They're still related to their mother cell or mother protein or mother sequence, but they are not identical, and they are also not related to each other. There's distance here, right? Because now there's two mutations that separate them, not just one, as is the case with the root sequence. And then this trend continues, all right? So now after another round of replication, you got two sets of mutations here, two sets of mutations here. These are both conserved, all right? And then if you drop down, you got two sets of mutations here, two sets of mutations here. And the G's are both conserved, but these are different, right? And then if you start comparing the progeny across, right, you have a C and an A here, you have an A and a G there. So they become more and more divergent. And by looking at much, much longer sequences than this, right, like thousands and thousands of base pairs, you can start to unravel this evolutionary relationship between different species based on these really conserved proteins. Because even conserved proteins still acquire mutations randomly because of the intrinsic error in the DNA replication machinery. Are there any questions about this concept? All right. Well, just in case there are, and you're still wrapping your head around it, I've created one last little thing. Ice cream. Everyone loves ice cream. If you don't love ice cream, please see me after class. All right. It's just like the beginning was a scoop of ice cream. Someone was like, I'm going to invent something today. And they invented a scoop of vanilla ice cream. It never existed before. Like, wow, nice. And then there was like time that elapsed and a mutation occurred. And it's like, boom, now we're going to add a bowl. Amazing, because who wants to eat ice cream that's sitting on the ground? But in the replication of ice cream, there's another mutation that occurred, which was a cone. It's like someone came along, was like, hey, you want to know how to like make children sad? You put something they love on a very precarious device and then watch it fall. Like, who invented the cone? Absolutely not. Bowl every time. I said it. Okay. Then you do it again, right? Shoop. Someone was like, what if we made two scoops in a bowl? Genius. Absolutely genius. But then someone was like, what if we just added a scoop, but brought in this thing called chocolate, which, as we know, is the best thing ever. And then people just like passed out. They're like, oh my gosh, like mind blown 1,000 times. Right. So like that's like where I would be in this like life cycle is like, oh, and then like Dr. Adams, like his family lineage like emerged there. Like that's where I'd be. And then there's like people over here in Crazy Town who are like, oh, we're gonna like keep expanding on the ice cream cone and like add a scoop on top that's a different flavor. Is like, okay, like congratulations, it's gonna fall. You're gonna be sad. I don't know why you're like doing this. And then there's some like real like wild things that are like this. Like I was Googling a lot of ice cream images to like make this slide for the class. And this is what I like. I did not know this existed. I don't know if it should exist. I don't know, like, I don't know what happened, but it definitely, like, of all the mutation visions of a mutation for an ice cream cone, this looks like a mutation. Like, I don't know what happened. If anyone has had one of these, please let me know, because, like, that's wild. Like, that's like, don't let that get out, because it could cause like a weird cone pandemic or something. All right. So basically, I made this whole ice cream analogy because each ice cream generation corresponds to the generations down here. Okay. So I've given you 3 different flavors of understanding this: the boring science way, the cool picture way, or the super cool ice cream way. So if I ask you a question about it, I think you have a chance. Are there any questions about this concept? All right. Well, I hope you're ready for a question then. My turn. So, an unlikely sequence of events, or likely, right? So like, it was sort of like a pun, not really, but it's fine. I gave you ice cream. All right. So you are studying protein sequences in pictorial form. Okay. Which option places them in the most likely order from the original sequence to the last sequence? So instead of having you stare at a bunch of base pairs, which might hurt your eyes, like it hurts mine, I'm giving you pictures. You're welcome. Thank you. So take a look at the answers. If you're like, oh snap, I'm confused, it's okay. That's why we have these questions now, to see where you're at, and then we can resolve any confusion. Your time starts now. Sorry, I've been watching a lot of Top Chef lately. No spoilers, please. And like, they always do that for the challenges. They're like, your time starts now. Alrighty, responses are coming in. Give everyone another, let's do 10 seconds. All right. Last call. Here we go. All right. A lot are at C. I see you're right. Yes, that is correct. If you did not get C, look carefully, right? You can just sort of navigate the most likely outcome, right? Because this sequence allows you to acquire 1 mutation at a time, whereas the others either start with a lot, drop off, then gain some, or gain 1 but then lose some, then go gain a lot, right? So C is the correct answer. And conceptually, like, this is how mutation rates and molecular clocks are grounded, except they're doing it with DNA base pairs. Let's go. All right, so we're now moving on to some related concepts that talk about vertical and horizontal gene transfer. Okay. So basically, we have a traditional view of how gene transfer works, right? So we've talked about mutation rates and how that can help us understand relationships. Now we're talking about how genes can be passed down to different species. And so the traditional view posits that the majority of gene transfer is vertical. So it just comes from the parents of the organ. The daughter cells get the majority of their genes from their mother cell. However, an alternative view argues that horizontal genes transfer is so prevalent that there are no clear lineages anymore. So transfer of genes from offspring in the same generation, for example, through plasmid transfer or the transformosome, we talked about that with strep pneumo, right? So it integrates free extracellular DNA. So they're saying that that happens so frequently that it actually really distorts these clear lineages, and there is no uh, dominant lineage hierarchy because this horizontal gene transfer is so pervasive. Okay, and so how we can explore this concept further is by first thinking about the different kinds of genes that exist in organisms, and they can broadly be defined as either informational genes or operational genes. So informational genes are what encode the essential products for transcription and translation. And because they're just so fundamental to the survival of an organism, they tend to be transferred vertically. And they also interface with so many different components that just getting it horizontally transferred would be really difficult to just plug in, to a new organism. The other group, operational genes, govern a lot of relatively important processes, but they're more flexible, right? It's not required for the transcription and translation of the genetic material in your organism. Instead, there are things like, oh, I can eat a new nutrient, great, like, or I can I can navigate this new stressful environment, or oh, I can like fight off reactive oxygen species when I am being attacked by white blood cell, right? So these different genes are sort of like tools or like upgrades to your bacteria or organism of choice. And so you can sort of explore this a little deeper by thinking about how different species may acquire or lose different gene subsets. So for example, the textbook looks specifically at E. coli and Salmonella enterica, and these have a last common ancestor. However, because different genes were lost and gained over many, many generations, they have since diverged into separate species. But there are a lot of similarities, both with regards to the weapons that they have, as well as the habitats that they colonize. And so from a large overview perspective, this kind of analysis can, again, show you why certain species have similar strategies in exploiting the host environment or metabolizing certain nutrients. And so this, I think, ties in nicely with our upcoming units on microbial pathogenesis, because there are different forces at play that push organisms to survive and in some cases thrive in these different habitats. And this is based on the principle of natural selection. So if we have these forces at play, these evolutionary forces that are on the scales of thousands and thousands of years, how can we study this? How can we truly decipher the complex evolutionary relationships between species? Well, fortunately, we have several types of evidence. This includes genomic analysis, strongly selective environments, as well as experimental evolution. hashtag scientist, no big deal, okay? And so one approach is the genomic analysis, right? So now all of a sudden we're comparing sequences that are both within and between genomes that can give us insight into not only how related organisms are, but also how they survived in a sometimes very different environment to what we currently experience, right? So if we see that certain genes were not present in an organism thousands of years ago, then it's like, oh, I guess that wasn't important, right? And then we can also pick up on when they became important, when those organisms acquired them, right? So there's lots of interesting um, information that can be gleaned when we look at these different sequences. We also can find events such as gene duplication, which provides the opportunity for paralogous genes or paralogs to evolve into different functions, right? So if you have a duplication event, then the duplicate gene, because it's redundant, could be mutated and eventually acquire a completely different function. And as we briefly talked about last week, there are also incidents of degenerative or reductive evolution when you no longer need a certain gene. Maybe the environment has changed, or maybe your nutrient acquisition has improved. And so certain genes can be lost or degraded because they're no longer important. So to reflect on this information, if you are a bacterium, all right, so let's all get our bacteria hats on. What single trait or property would you have to make you the most fit? Okay, so you can choose anything out there, but you can only choose one thing. You want antibiotic resistance, you want to be able to grow really fast, you want to be able to swim really fast. Like, what would you choose? Just drop it in the chat. And remember, the goal is to be the most fit. Free access to the gym. Don't go to the gym right now. Still in a pandemic. What would you choose? I have a direct message for fast growth, fast replication, resistance for show. Oh, all right, some antibiotic resistance fans out there. Okay. Anyone else? All right, fast spreader. Okay, I like that. Well, all of those options are good, but ultimately it's going to depend on your environment. Deadly, brutal. All right, and so I think one of my favorite little vignettes of the book is when they looked at methicillin resistant Staphylococcus aureus. Okay, so this bacterium, which we'll be talking about in a week or so, was initially sensitive to 4 antibiotics. All right, so someone came in with an infection and then they cultured it, and it was basically sensitive to 4 different antibiotics that are used to treat this infection. And then after 12 weeks, it had begun to become resistant to all 4 of them. Okay. So it's sort of like scary how resilient bacteria can be, right? And that's why MRSA is such a dangerous bacterium, right? You have our best antibiotics doing nothing against it. And we're not generating that many more antibiotics, which is a problem because we are going to quickly reach a point unless we make some very dramatic changes in how we use antibiotics. And fight bacteria and microbes at large, the strong selective pressures replacing these bacteria under are coming back to bite us because we're running out of ways to defeat them. Okay. And so this is just one example of how these selective pressures can drive bacteria to acquire mutations or genes that will make them resistant and thus propagate further to become a dominant population in our own ecosystem. And so ultimately the fittest trait depends on the environment in which the selection occurs. So while you have this example of antibiotic resistance being extremely important, you could have another environment where maybe there's tons of reactive oxygen species, and that having mechanisms to protect yourself from reactive oxygen species could be really important. And so if you remember way back at the beginning of class, we talked about some of those different strategies where you try to detoxify or neutralize reactive oxygen species. So ultimately it's going to depend on the environmental pressures that you face as an organism. And so to study this, right, it's good to not just take clinical samples and be like, all right, well, this person's almost dead. I guess we should try to study this, right? We want to try and get ahead of the game here, right? And so that's something we do with experimental evolution in the lab. And you're like, isn't that redundant? Everything's like an experiment in the lab. It's like, look, it's experimental evolution, all right? We're going forward in time. This is sort of like the closest thing we have to a bacterial time machine. And so basically how these experiments work is they will create a population of bacteria that is then grown under a certain set of environmental conditions, right? It all goes back to your environment. We're products of our environment, right? And so this initial population is then going to be grown in a liquid medium, and then the culture is going to be diluted, and then you just propagate it over and over and over again. And next week, we will talk about this great long-term evolution experiment, or LTEE, that has gone on for just, I think, over 60,000 generations. And if you want to know like super nerdy vibes, I mean, look at this image. Like that is a ton of Petri plates. So you've, I think, all struck out bacteria now. Like that's thousands of plates, thousands. And we're going to dig into the cool things that you can learn from these experiments. And it's not just about building gigantic Petri plate sculptures. All right, that is a wrap for today. I mean, it's not a wrap, like, you know, I'm not busting a flow right now, but it